All right, it's time for active transport. This is the last type of transport across the membrane. It's any transport that requires energy because the cells are pretty much pushing the materials against the concentration gradient. So we're going to be talking about situations in which the cell is carrying things against the concentration gradient. So that's active transport. Now, active transport requires energy, which in terms of molecular energy is ATP. All right, so this usually happens either through proteins or through changes in the cell membrane structure itself to create the system. There's basically two types of active transport. There's massive bulk active transport and small active transport. So this is an example of active transport. So you have more things on the inside than on the outside. So it should go towards the outside. But I want that molecule to come in. So I need to spend energy in order to pump that molecule through the membrane to the end. Example of a transport. It's going against the gradient. The transport moves things from low to high concentration, or in other words, against the concentration gradient. And a very good example of this is the sodium-potassium pump. There's a very good web tutorial about it on my website. But basically, the sodium-potassium pump is a pump where the potassium is pumped in and the sodium is pumped out all at the same time, or vice versa. And so it looks kind of like this. I'm sure so you're going to have the channel protein like that, and that's getting across the membrane, right? And then you're going to have the sodium being direction, and meanwhile you're getting the potassium pumped in the opposite direction. And both of them will be requiring ATP for that to be happening. And so each time a sodium or two potassiums are being pumped across the membrane, it takes ATP to do that. And that's actually very important because this is how neurons create the shock, the action potential that sends a message from one end of the nerve to the other. So when your brain wants to send a message from here down the spinal cord, and then from the spinal cord to your fingertips, it goes through electrical impulses, and those impulses are generated by the static charges, which have everything to do with the potential of sodium and potassium. So by controlling the amount of potassium in and out of the cell creates a charge differential, which sets that charge. So it's very important for your body. All right, sodium-potassium pump. Now, here you go, it's showing you that the movement of one sodium or two or three potassiums every time, and this will be happening against the gradation, the energy. So very good. Now, another example of that, oh, by the way, I don't have a video about that, but we will soon be talking about cellular respiration and photosynthesis. And when we do that, you're going to go over the idea of a proton pump, which is exactly the same thing. It's pumping protons against the concentration gradient. Basically, it's charged hydrogen atoms. And this is going to be very important in the mitochondria, the cell membrane, in the chloroplasts, which are all part of the cellular energetics process. So you have action potentials in neurons and cell respiration and photosynthesis both requiring active transport. So definitely very important for the membrane. Another example of active transport that I want to talk about before I talk about this moving the big stuff is an example of Cotransport. Now, one way to actually do this without spending too much ATP is, let's say, for example, I have a lot of certain things outside the cell, and I want to get this is going naturally this way, right, because of diffusion. So if I put, say, a gate here that has to be spun every time one of these molecules come through, like I call it a turbine, that can capture the energy that's from the gradient, the potential energy, into kinetic energy. Now, that energy can produce energy necessary to move another molecule the opposite way. So now, this molecule should definitely also be inside because there's a lot more of them in the inside. But if I want to pump this outside, I can use the energy that I gain by moving that molecule in to move this molecule out against its concentration gradient. So this is an example of cotransport that it happens sometimes in cells. So to save ATP, Now, to move the big stuff, we have two processes. One of them is called exocytosis, and the other one is called endocytosis. Now, exocytosis is moving things out of the cell. So as you can see here, you have all these little proteins inside a transport vesicle. Now, what this vesicle will do is that it's made of fat. The protein is polar, so it wouldn't get to the membrane. But if I envelop that protein in a layer of fat, what happens? That fat can merge with the cell membrane and then allow the protein to exit into the extracellular fluid. So that's actually very interesting. And remember, what is actually doing that? What is packaging the proteins in this layer of fat? That's the Golgi apparatus. Remember, we talked about that. The Golgi apparatus is going to be the thing that's going to be pushing or enveloping these proteins into that. So when we said that the Golgi apparatus was a packaging machine, it does that. But remember that the Golgi also packages things to be sent within the cell. So it organizes what goes what inside the cell. But okay, so here you have an example that's showing you an actual picture of exocytosis, and you can see the exocytotic vesicle as it merges with the plasma membrane and releases all those proteins from the inside of the cell. So this is an example actually of a cell that produced antibodies and is releasing those antibodies in the cellular medium. So a good example of that. Here's another example that shows you how the actual vesicle is going to be merging with the membrane. And I really like that graph because it actually shows you that the vesicles themselves are made just like the membrane of a lipid bilayer, which allows them to integrate with the membrane. So if you think about it, this is the same process that builds the membrane itself. So when the cell wants to grow, it grows by incorporating vesicles into the membrane. So this is a good example of that. So remember, the Golgi is also in charge of enlarging the cell in addition to all the other jobs I just mentioned. Now, this is inside the cell, and that's the outside the cell, of course. Now, there's another way of moving the big stuff, which is endocytosis. So you see here an example, a sequential example of a cell membrane in an electron microscope picture that's folding inwards to create a vesicle. So this is actually the opposite of the exocytosis process we just saw. Now, this called endocytosis, which makes an endocyte, which is basically the name for that vesicle that comes off the membrane. And as you can see, all those little particles which were attached to the cell membrane are now incorporated inside the vesicle, and that's going to go into the cell. Now, two examples of endocytosis are pinocytosis. The first example of pinocytosis, I know it's a strange name, but you have these proteins, you want to bring them on the inside. So this is also referred to as cell eating. It's the most common form of endocytosis. The cell basically just closes up the cell membrane, as you can see in the picture, and creates this vesicle. All right, endocyte. Now, this endocyte will go ahead and merge with a lysosome, and the lysosome will do the job of digesting whatever those things are, so that the cell can get the basic parts of that and use it for whatever it wants to use. So remember, that's how the process works. Now, this is only going to work to get things which are dissolved in molecules in the liquid that's outside the cell. Now, another example of pinocytosis is the idea that this happens through an invagination. Invagination is the shape, the V shape that creates on the cell through the process. So you can see an example of that here. I know pinocytosis, invagination, but these names are basically invagination. Basically that's what it means. But invagination means even got confused there. It means basically a protrusion or a hole in the cell membrane. All right? In pinocytosis, pino basically means drinking. So that's an example of a cell drinking because it's getting the stuff that's dissolved in the cellular medium. Here you can see an example of that actually happening with an electron microscope, and you can see vesicles at several stages of their development. Right? Very nice. Now, this is actually happening across a capillary cell in the blood, all right? So cells enveloping stuff. Now, a specialized example of pinocytosis is, what if I don't want everything that's in that liquid to come in? What if I want just certain things to come in? So what the cell does is it uses another cell component that I forgot to mention when I did the first cell membrane component lecture, which is a glycolipid. A glycolipid is basically a fat that has some carbohydrates attached to it, and this fat, these little fat-looking things, will attach to a specific molecule, all right, like a lock and a key, and once they are attached, it induces an invagination in the membrane, which will create a vesicle that is sucking in only the things which you want inside. So it's a way to receive those things. It could happen. This is how the cell takes in hormones and other things like that into the cell. So again, this is a really good example, the same picture you saw before, of receptor-mediated phagocytosis. Now, another big example is phagocytosis. Now, phagocytosis, instead of creating an endocyte, it will create a phagosome. Phagosome is just another name for a big endocyte, which will merge again with the lysosome, and just like before, it will form a food vacuole where the lysosomes will be working and digesting those things. But this is how the cells eat ginormous things, like enormous material. And this is different from an invagination. You're not going to just create a little vacuum for things to get through there. The entire cell membrane has to protrude and hug the material that it wants to eat, all right? And engulf that, So this is how big particles of food and bacteria that there are in the blood or blood cells are trying to defend against are actually engulfed, So here's an example of that. You see the little bacteria there about to be engulfed by a white blood cell called a phagocyte, that is going to be enveloping that and then nuking it with lysosomes. So this is a good example of phagocytosis. And here's another example of a yeast cell being captured by our immune system. So the phagocyte trying to envelop the whole yeast cell. On the web tutorials, there's actual videos of this happening with electron microscopes. Now, that is basically what I have to say. So remember again that you have two types of transport, all right? So you have two basic types of transport. You have the active transport and the passive transport. On the passive transport, you have diffusion and facilitated diffusion. Under active transport, you have small active transport and large active transport, right? Small active transport examples like potassium pump and hydrogen pumps. This large transport, you have endocytosis and exocytosis. For the endocytosis, you have pinocytosis, receptor-mediated pinocytosis, and phagocytosis. For the diffusion, you have facilitated diffusion and simple diffusion. And the diffusion, examples of oxygen, carbon dioxide, things like that, and fat. For facilitated diffusion, you have things like sugars, amino acids, and proteins going through channels or carrier proteins. So, and water too, which goes to the aquaporins. And remember that a special example of facilitated diffusion is osmosis. So that, in a nutshell, is transport across the cell membrane, and I hope these videos were helpful. Come to class, understanding these concepts, watch Bozeman Biology, watch the web tutorials, make sure you do everything you can to come to class understanding these concepts so that we can just do labs and apply them, all right? Thank you very much, and don't do anything that would make your mom proud.